So our final presenter today is Bhedita Seewoo, and the title of her talk is Combined RTMS MRI Studies in Animal Models to Study Lasting Effects of RTMS. So Bhedita is a fourth-year PhD candidate in neuroscience at the University of Western Australia, and Bhedita's research focuses on the use of in vivo MRI to examine the effects of low-intensity RTMS on brain activity, chemistry, and structure in rodent models to inform protocol development for human studies. So whenever you're ready, Bhedita, please take it away.
All right, thank you everyone for giving me the opportunity to present today. It's really great. So for people who are in the human MRI and RTMS field, I would hope that at the end of this talk, I can convince you all that doing animal model works in the animal model is important. And for those who already work on animals, hopefully I will be able to convince you that doing neuroimaging in animal models is important too. So let's get into the talk. As we have heard from previous speakers, MRI studies in humans have considerably increased our knowledge regarding the potential mechanisms of RTMS. However, subject-related variables such as gender, age, exercise, diet, and genetic background might affect the stimulation-induced effects of RTMS in both healthy individuals and patient populations. To improve the safety and efficacy of RTMS in a clinical setting, a better understanding of RTMS is required. So animal models give us the opportunity to reduce variability in RTMS effects by controlling for many of these factors. For example, we can perform experiments on animals which are of the same gender and age, and at the same time ensure that they have the same diet and are from the same genetic background. We can even pick female rats, for example, which are genetically bred to show high anxiety and study the effects of RTMS in them. Being able to understand the effects of RTMS in such populations will drive personalized RTMS treatments in humans, as Andrew Zalewski has talked about a little bit before. So because animals introduce considerably less variability in experiments, they have been useful in elucidating some of the important mechanisms of RTMS. The benefit of animal studies is that it gives us the opportunity to perform invasive studies of molecular and genetic changes that are not possible ethically in humans. For example, previous animal studies have shown that RTMS can induce the expression of plasticity-related genes, it can result in upregulation of growth factor expression, and it can also induce neurogenesis. Despite the fact that RTMS is being used in a clinical setting and that animal models are increasingly identifying potential cellular and molecular mechanisms, little is known about how these mechanisms relate to the clinical changes that we see. This knowledge gap, I think, is further amplified by non-overlapping approaches using clinical and preclinical studies. Clinical studies are non-invasive, of course, using MRI-based techniques, as we have seen from previous speakers, while preclinical studies are mostly invasive and are performed post-mortem. A non-invasive method is therefore needed in rodents to link the understanding of cellular and molecular changes in the brain to brain changes that are clinically relevant. In other words, MRI can be performed at several time points during an RTMS study in animals, and post-mortem data can be acquired at the end of the experiments using the same animals. And then if we use the same detection tool, we will increase the translatability of animal studies, as MRI can act as a bridge between what we see in post-mortem data and what we see in non-invasive human studies. So I would say MRI is a technique of choice for measuring changes in the brain non-invasively so that they're comparable to human studies, because most human studies use MRI methods. Different MRI sequences can be used to obtain different types of information using the same scanner. So for example, if you want to look at functional changes in the brain, you can use resting state functional MRI. And this method measures brain activity when the subject is at rest. You can also look for chemical changes in the brain using proton magnetic resonance spectroscopy, or MRS. This technique allows for the detection of in vivo cerebral metabolite levels. You can look at structural changes in the brain using anatomical imaging. So you can just simply extract the volume of regions of interest from the brain and look at changes in volume that way. Or you can do diffusion imaging, which maps the brain's white matter tracks. So in my studies, I've used several of these methods. In the last one that I will talk about, I've used actually all of them. So just for you to know what I'm going to talk about. So first of all, I'm going to talk about some important considerations for using RTMS and resting state functional MRI in animals, because it's not as direct, it's not as obvious. as human studies. And then I'll talk about some of my studies. So this one was from my honors, and I did like a quick study where I looked at immediate changes in following one session of rTMS in animals, in healthy rats. And then I did a more longitudinal study where I looked at changes following repeated rTMS delivery in healthy rats again. And then we wanted to use an animal model of depression, but before looking at the effect of TMS in that model, we had to validate that model first to see if the animals are showing similar MRI-related changes as seen in humans with depression. And then we tested the effect of rTMS in that model. So first of all, the considerations for doing resting state functional MRI and rTMS studies in animal studies. So a bit of background on resting state fMRI first. fMRI measures brain activity by detecting changes in blood oxygenation. So deoxyhemoglobin being paramagnetic decreases signal intensity, which creates a contrast between the blood vessel and the surrounding tissue. This contrast can be used to monitor blood oxygenation in different parts of the brain based on metabolism or blood flow. An increase in brain activity increases cerebral blood flow. This increase is, however, greater than the increase in demand and hence supply of oxygen to neurons. Therefore, there is an excess of oxygen in the brain when it is active compared to baseline, even though the use of oxygen has gone up. So resting state functional MRI detects functionally linked brain regions with synchronous activity. These brain regions form an organized network called the resting state network. The default mode network is one example of a resting state network. So the image on the left here shows a human default mode network. The yellow parts of the brain are all active together. So human studies have shown that patients with neuropsychiatric conditions have disrupted resting state networks, and resting state functional MRI can be used to track improvements in functional connectivity following rTMS. Despite the distinct evolutionary path of the primate and rodent brains, several studies have confirmed that humans and rodents have very similar resting state networks. So this is a rat default mode network. It looks quite different visually, but similar brain regions are active in both species. So one example is the cingulate cortex highlighted there, which is an important part of the default mode network, but we'll come back to that a little bit later. So because of this similarity, measuring rTMS-induced changes in the resting state network in rodents would be clinically relevant. But we have another issue. The use of anesthesia is generally required in animal MRI studies in order to immobilize the animal and reduce stress. It is known that anesthetics alter the neurovascular coupling, but as we saw before, fMRI signals rely entirely on the coupling between local blood flow and local neuronal activity. As such, the use of anesthesia may confound imaging results. Different anesthetics have different effects on the cerebral blood flow, and therefore fMRI signals are altered in an anesthetic and dose-dependent manner. So a study by Passanen et al. In 2018 compared the resting state data from awake rats to rats scanned under the influence of 6 different anesthetic protocols. Just a quick note, it is not common to do studies on awake rats as they require months of training to prevent stress during imaging. So the top row here depicts what brain regions were active during imaging in the awake rat, and the other 7 rows are difference plots. So they depict how different the brain activity were compared to the brain activity when the rat was awake. So fMRI signals are considered accurate when they produce an image reflective of brain activity in the awake state. Therefore, we don't want any colors in the difference plots. As you can see here, isoflurane, which is a vasodilator, causes an apparent increase in activity. And we have medetomidine, which is a vasoconstrictor, and it causes an apparent decrease in activity, which is depicted by the blue colors. We also have alpha colorose and urethane as well, which looks like they might be good ones, but they can't really be used for longitudinal studies. Alpha colorose causes liver toxicity, and urethane is a carcinogenic. So, but a combination of isoflurane and medetomidine shows minimal differences from the awake condition. So this is the anesthetic that I use in my studies. It produces similar resting state connectivity as in the awake condition, provides stable sedation for over 4 hours, and it also produces reproducible data from repeated fMRI experiments on the same animals carried out 1 week apart. So briefly, the animal is first placed in an induction box where it receives 4% isoflurane, After about 6 minutes, it is transferred to the MRI bed where the nose cone delivers 2% isoflurane, and you allow the animal to stabilize for some time. And then medetomidine is delivered subcutaneously using a butterfly catheter into the left flank of the animal. So we deliver a bolus and continuous infusion of medetomidine throughout the experiment. So once you deliver the bolus of medetomidine, you slowly reduce the isoflurane concentration to about 0.5% while maintaining the breathing rate of the animal between 50 to 80 breaths per minute. So fMRI data, I would say, should only be acquired once the animal stabilized on a low isoflurane concentration, so about 0.5%. The second issue now, when we're doing rTMS studies in animals, is to do with the size of the coil. So if you have a human figure of 8 coil, it would stimulate the brain quite focally. But if you use the same coil in animals, it would stimulate the whole brain, if not even part of the body of the animal. So we need smaller coils. So our lab has developed custom rodent coils, which are circular coils, and they can focally deliver stimulation to small animals to about like one hemisphere of the brain. But if you have smaller coils, then you have an issue with the intensity of stimulation that you're delivering. So if you have a human brain and you're delivering about 1 to 2.5 Tesla of stimulation at 100% motor threshold in humans, the distance between the coil and the cortex is about 25 millimeter, and you get about 80% attenuation in signal. So by the time the signal reaches the cortex, you're actually receiving 0.2 to 0.5 Tesla of stimulation. In contrast, for rats, if you deliver stimulation at 0.02 Tesla, which is about 15% of the motor threshold in a rat, the distance between the coil and the cortex is only 2 millimeter. Therefore, you get only 10% attenuation in signal, and the rat is actually receiving 0.018 Tesla of stimulation at the cortex. So even though you're starting off 100 times with 100 times less intensity, the brain is actually receiving only 10 times less in intensity of signal. So for my studies, I used one of these miniaturized rodent coils with the specifications shown on the slide. It delivers an intensity of approximately 13 millitesla at the surface of the cortex. As you can see on this image here, mapping the magnetic field of this coil, the custom-built round coil that I use in my studies delivers stimulation focally to only one brain hemisphere. So using this coil and using the anesthetic method that I've described, we wanted to see if we can detect the effect of a single stimulation session using resting state fMRI in healthy rats. So we did a quick pilot study using 6 animals. First, we used a baseline scan to identify the resting state network in our rats. For those who are not familiar with rat brain images, so if that's a rat and this is a rat brain, if you slice it this way, you get the top coronal image, and if you slice it like that, you get the bottom axial image, and the colors show functional connectivity. So stimulation was delivered at 4 different frequencies, and MRI data was acquired immediately before and after the 10 minutes of stimulation. And as you can see on the images, the functional connectivity of the networks are affected quite differently following each stimulation type. I won't go into a lot of detail about what we found, but if you look at the 10 hertz, for example, you can see right where the stimulation was delivered, there's a decrease in functional connectivity of the somatosensory cortex and the motor cortex, and the changes are dependent on the frequency of stimulation that we used. So then now that we know what happens following a single stimulation session, we wanted to investigate the longitudinal effects of repeated rTMS delivery. So in this study, we acquired information about the baseline resting state activity by scanning the animals on day 0. The animals then received 10 minutes of low intensity rTMS on the right side of the brain at 10 hertz or 1 hertz for 15 days. We stopped stimulating the animals and then imaged them on day 14. We were very interested in finding out what happens after the cessation of stimulation. So another advantage of performing animal studies is that animals are always able to come back for follow-up time points, which is something quite challenging in human studies. So we also imaged the animals on day 21 and day 34 for the 10 hertz group, and on day 21 and 28 for the 1 hertz group. The only reason why the last imaging time point was different was because the MRI needed repair for a few days, so we just had to wait, and there was nothing we could do. So what we found was quite interesting. For the 10 Hz group, we found that connectivity to the basal ganglion network increased after 14 days of stimulation. These changes were seen in the contralateral hemisphere. So the contralateral hippocampus, sensory and striatum were functional connectivity were increased. So this is interesting because, as you might remember, the magnetic field of the coil that I use only reaches the stimulated hemisphere, as the human coil does in humans. But we know from RTMS studies in humans that RTMS can induce changes both locally and also in the remote brain regions. So this wasn't as surprising, I would say. After cessation of stimulation, functional connectivity decreased compared to day 14. As you can see in the second image at the top, when comparing functional connectivity on day 14 to day 21, we see that functional connectivity within the contralateral hippocampus is only slightly lower on day 21 than day 14. This is demonstrated by the redness of the color in that brain region. Greater changes are more yellow. At this time point, functional connectivity within the primary somatosensory cortex is still quite high and therefore doesn't show up as being significantly different to day 14. After 13 more days of no stimulation, in the last image at the top, the hippocampus is more yellow now, showing that functional connectivity has decreased further compared to day 14. And now even the primary somatosensory cortex has lower connectivity than day 14. All of that to say that after cessation of stimulation, functional connectivity gradually decreased over a period of 20 days in the 10 Hz group. In the 1 Hz group, we saw sort of the opposite. So there was a decrease in functional connectivity following 14 days of stimulation. And after 7 days of no stimulation, functional connectivity was still lower than baseline, which means that it was lasting quite a long time. But by 14 days of no stimulation, the functional connectivity went back to baseline levels, so we didn't see any significant difference there. The main findings from this study is that we found an increase in functional connectivity with a 10 Hz stimulation, but a decrease with 1 Hz stimulation. Importantly, these changes were similar to MRI-related changes that we see in humans following brain stimulation. Importantly, there was more widespread changes with the 10 Hz group than the 1 Hz group, but the changes seen in connectivity were sustained for longer in the 1 Hz than the 10 Hz group. So now that we know how RTMS alters the brain of healthy animals, the next step for us was to investigate the effects of RTMS in an animal model of a neuropsychiatric condition. So we chose to study an animal model of depression. There are more than 300 million people with depression worldwide, and it is estimated that 1 in 7 Australians will experience this disorder at some point during their lifetime. There has been a lot of research in this disorder, and it is now generally accepted that depression is a complex disorder. It is mostly diagnosed through changes in mood and behavior, but people with depression also exhibit changes in brain function, chemistry, and structure. Animal models have played a key role in the testing of the safety and efficacy of treatment of depression. One such animal model is a chronic restraint stress model. It involves placing the animals in individual transparent acrylic tubes to restrict their motion, as you can see on the image on the left. This is usually done for about 2.5 hours daily for 13 consecutive days. The continuous and predictable nature of this stress is designed to mimic the everyday stress that people experience, such as having a stressful job or familial stresses. Studies have shown that following restraint, the animals exhibit similar genetic and protein changes that are seen in humans with depression. But what about the functional, chemical, and structural brain changes that are seen in humans with depression? This is what this first part of the study was about. So before we can test the effect of RTMS using MRI, we had to validate this model first to see whether the animals were exhibiting these changes at all. So in order to test this, I used 33 Sprague Dawley rats. 25 of these animals were subjected to the restraint procedure, while the 8 healthy controls were left within the cages during that procedure, but still underwent all the tests. We performed behavioral tests before and after the 13 days of restraint to confirm the presence of depression and anxiety-like behaviors in the animals, because you can't really ask a rat how it's feeling. And we also performed MRI before and after to detect the effect, to detect the brain changes. So the first behavioral test that we did is called the elevated plus maze test. Following restraint, the animals show less exploration in general within the maze, but specifically, they're afraid to explore the open arms, and therefore they stay within the closed arms for longer. The second behavioral test we did is the force swim test. It involves placing the rats in individual buckets of water. Following restraint, the animals show less climbing behavior, so they're not actively trying to escape anymore, but instead they exhibit learned helplessness, and they stay immobile, floating in the water for longer. So both of these tests confirm the presence of depression and anxiety-like behaviors in our animals receiving restraint, while the healthy controls did not show any changes in behavior. In terms of analyzing the MRI data, we simply compared the baseline data to the post-restraint data. The changes in functional connectivity of the cingulate cortex was determined using resting state functional MRI. The cingulate cortex is a critical element of the default mode network, as I mentioned before, and it has been extensively studied in the context of depression. This network is implicated in rumination, self-referential functions, and episodic memory retrieval. We found a significant increase in connectivity of the cingulate cortex to several brain regions highlighted in this image here. Healthy animals did not exhibit any changes in functional connectivity. The results of the current study is consistent with human studies showing evidence of increased connectivity between the cingulate cortex and other limbic areas in depression. The increased functional connectivity of the cingulate cortex is believed to be associated with increased emotional processing at the cost of executive function. We also acquired magnetic resonance spectroscopy data within the sensory motor cortex highlighted in blue, and we found a significant decrease in glutamine and combined glutamate and glutamine levels following restraint, but not in healthy controls. And this as well is in accordance with previous preclinical and clinical studies, which have proposed that an altered glutamatergic neurotransmission plays a pivotal role in the pathogenesis of mood disorders. We also looked for volumetric changes in the brain. So the hippocampus highlighted in red was extracted from the T2-Weighted anatomical MRI images, and we found a significant decrease in hippocampal volume following restraint. There are no changes in hippocampal volume in the healthy controls. So a reduction in hippocampal volume has also been consistently associated with depression in humans, and it's believed that cumulative exposure to disrupted emotional regulation, stress reactivity, and glucocorticoids as a result of depression is believed to increase neuronal susceptibility to insults and therefore lead to hippocampal deficits. So diffusion MRI was also used to investigate structural changes in the brain, but within the white matter. It provides a quantitative measure of white matter fiber morphology and fiber density. So you can measure the cross-sectional size of a fiber bundle. An increase in fiber cross-section would look like the image on the left. On the other hand, apparent fiber density reflects the number of axons or axonal size. So an increase in fiber density may be due to greater number of axons, as you can see here, but it can also be due to larger axons just occupying more volume. An increase in fiber cross-section and fiber density is usually considered a positive change because it is known to occur in brain development and repair. But when we looked at our data, we found a significant increase in fiber cross-section and fiber density in both the healthy and restrained animals, which was a bit confusing at first, but previous animal studies have shown that cerebral development occurs in rats until they are about 6 months old. Our rats were around 8 weeks old at the time of the baseline scan, so it would make sense that it would still be exhibiting brain maturation-related changes. So because both the restrained and healthy animals showed changes, we wanted to quantify the amount of change with and without restraint, and therefore we compared the baseline time point to the post-restraint time point using an equal number of animals in the healthy and restrained groups. So the healthy animals showed a considerably greater increase in fiber cross-section and apparent fiber density compared to animals which actually underwent restraint. The colored regions in these brains show an increase in the parameter being tested. So as you can see, for example, healthy controls showed an increase in fiber cross-section in voxels adding up to 134 millimeter cube, while the restrained animals show an increase in only 31 millimeter cube. Therefore, animals subjected to restraint exhibit less brain maturation-related changes. This is an important finding because it shows that even with a short duration of stress, and in this case, it was just 2.5 hours daily for 13 days. If you have a short duration of stress in a young developing brain, this can have long-term consequences. So the main findings from this part of that depression study was that we found functional, chemical, and structural changes in the rat brain following restraint. And importantly, these results are similar to MRI-related changes observed in humans with depression. So we believe that this part of the study validates the model of depression, and validating this model of depression was important because animal models are an indispensable tool for studying the etiology, progression, and treatment of depression. But there's still controversy regarding the validity of using rodent models of human neuropsychiatric disorders because the behavioral tests that we do in animals are not directly comparable to the changes in human behavior that you see in depression. MRI findings, on the other hand, are more directly comparable. Therefore, I believe that to increase translatability of results, future animal studies can investigate the effect of new treatment methods on MRI measures in addition to measuring improvement in behavior. The next step was to determine the effects of stimulation on this model. We're still analyzing the data for the fMRI, MRS, and hippocampal volume with this, but I have some results from the diffusion data, which I can show you. So after restraint, the animals received an accelerated protocol of low-intensity RTMS delivered to the left dorsolateral prefrontal cortex. In the clinics, RTMS is usually delivered once daily for 4 to 6 weeks, but there's increasing evidence that receiving multiple stimulation sessions a day can increase efficacy of the treatment while also decreasing the treatment period. Therefore, we delivered stimulation to the animals for 10 minutes, 3 times daily, every weekday for 2 weeks. The stimulation sessions on each day were spaced 1 hour apart. After these 2 weeks, diffusion MRI data was acquired at the post-RTMS time point to investigate the cumulative effect of the treatment, and we then stopped stimulating the animals and imaged them weekly at the week 1 and week 2 follow-up time points. So these follow-up time points are important because they will give us information about how long the effects of RTMS would last for. In terms of animal groups, we had 3 different groups. So the active stimulation group actually received the stimulation, and the sham group received a sham procedure, during which the coil was just disconnected. So the sham group was used to control for handling during the RTMS procedure. We also had the restraint control group, which underwent restraint, but did not receive any extra handling during the treatment period. To determine the effect of RTMS on the white matter, we compared the post-restraint time point to the post-RTMS time point and week 1 and week 2 follow-up time points. So just a quick note that even though the sham and restraint control animals did not actually receive TMS, we would still refer to the imaging time points as post-RTMS and so on. So when we compared the different time points, we found significant changes in the fiber cross-section only. So this is what the results look like. There's a lot of images here, but don't worry, I'll walk you through the findings. The first row shows changes in the active group over time. The second is the sham group, and the third is the restraint control. As you can see, fiber cross-section increased with time. So if you look across, they increased with time in all the groups. However, there was a greater increase in fiber cross-section in the active group compared to the sham and the restraint control groups. So if you look at the top row, stimulation caused the fiber cross-section to increase rapidly between the post-RTMS and week 1 follow-up time points. And then this increase stabilized between the week 1 and week 2 follow-up time points. And you can see that with like only 2 millimeter cube of changes here, but then these two are pretty much similar. In contrast, the sham and restraint control groups show slower increase over time in fiber cross-section, and this may suggest that the effect of restraint is wearing off. But nevertheless, if you look at the week 2 follow-up time points, fiber cross-section is considerably higher in the active group compared to the sham and restraint control groups. So the active group showed increase in fiber cross-section in voxels adding up to 217 millimeter cube, while the sham and restraint control groups only show an increase in 103 and 59 millimeter cube. So this is in line with previous animal studies showing that high-frequency RTMS can have a neuroprotective effect and prevent neuron loss, and more importantly, promote neurogenesis. The main findings of this study was that there was compromised brain maturation-related changes in the white matter following restraint, but these changes were stably restored by active low-intensity RTMS, which may indicate that the RTMS might be promoting neurogenesis or having a long-term neuroprotective effect on the white matter of the brain. As a general summary, I want to say that the use of non-invasive imaging methods to investigate the effects of low-intensity RTMS in rodents allows for direct comparisons between rodent and human studies. We can also perform postmortem studies. For example, you can look at neurogenesis or perform track tracing in animals and then link these findings to the functional, chemical, and structural effects of RTMS that we found using MRI. So this will help deepen our understanding about the mechanism of action of low-intensity RTMS in psychiatric disorders. So we can perform these studies in different animal groups to drive personalized RTMS treatments in humans in the future. I would like to thank my supervisors and everyone else involved in this study. Thank you for listening, and I'm happy to take questions.
Thanks. Thanks, Bhedita. Another great talk, and really interesting to see the work that's being done from an animal perspective. I'm just conscious that we're running quite a bit over time. Should we, if we've got time, we could probably take 1 or 2 questions before we wrap up.
Yeah, I'm happy to take questions.
If anyone
Yeah, that was a fascinating talk and really comprehensive.
Yeah.
I actually had lots of questions, but I'll limit it to only one. And I think in the last study you showed quite widespread effects of change in basically across the entire white matter volume in fibre cross section.
Mm-hmm.
I wonder if you could comment on the potential mechanism. I think you mentioned neurogenesis, but given that this is in white matter, what could be driving these changes within the white matter volume?
So I think that it's only because of growth in the brain. So as I mentioned before, the rat brain maturation occurs until they're about 6 months old, and these animals, by the end of the study, they were about only, I would say, 3 months maybe maximum. So yeah, so they're still growing. And what I think it's showing is that when you do the restraint procedure, so when you induce depression in these rats, you're stopping that brain maturation process. And if you deliver the active rTMS, it's allowing for that brain maturation to occur in only the active group, but not in the other groups. So the other groups still have slower brain maturation.
Thanks. Yeah, really wonderful talk. It's impressive.
Yeah, it's a lot of data. It's a bit hard to go through all of them.
We've just got another question in the chat, Bhedita. Thanks for a very interesting talk. Are the animals anesthetized when receiving rTMS,
No,
other than during imaging?
So the first study, because when we're looking at only a single stimulation, the effect of a single stimulation session, that one, yes, they were, because we basically scanned the animals and then took them out of the MRI while they were still on the bed, stimulated them, and then put them back in because we wanted to look at the immediate effects. But for all the other studies, no, they were just active on our lap, just delivering stimulation to them.
Right. Does anyone else have any other questions? Okay, in that case, I think given time, we might wrap it up. So thank you, Bhedita, for that great talk. And thanks to all the presenters for giving really interesting talks today.